call for the next speaker, Dr. Moreira, who is a research scientist at the Fiocruz Institute in Brazil.
Good afternoon, everyone. I just want to thank you, the organizers, for inviting me to be here. It's an honor to be here at the Academy. And today I'm going to talk about when a bacterium fights arboviruses. Okay, so this is the world dengue distribution from 2010 to 2016. And you see that the more dark in the map, more dengue burden in the countries. There is an estimation that there is 390 million cases happening every year in 128 countries, and 3.9 billion people are at risk of contracting dengue. I want to show you some historical data from Brazil since 1990. And you can see that over years, the situation has become very critical, especially in 2015 with the introduction of Zika and chikungunya with dengue. Only in 2016, we had over 2 million cases of arboviruses in Brazil. So this study was done in 2016. And there is estimation of, with 2 million cases of arboviruses, there was 344 million euros spent to fight the mosquito, 81 million euros for diagnosis and treatment of patients, and 100 million euros in indirect costs, like days away from work. This is a very, very strong burden of the disease in the country. We have dengue in Brazil for more than 30 years, and every year the government produces campaigns and education campaigns for the population, but still the problem is there. Millions and millions of liters of insecticides are used every year, and because of that, there's a huge problem of mosquito resistance in many cities in Brazil. We need urgently more tractful and also sustainable strategy for controlling vector-borne diseases. So this strategy is based on Wolbachia, which is...
Our method uses naturally occurring bacteria called Wolbachia that live inside insect cells and are passed from one generation to the next through the insect's eggs. Wolbachia are found naturally in up to 60% of all the different species of insects. Insects that include fruit flies, moths, dragonflies, and butterflies. The secret to the success of Wolbachia is how it manipulates the reproduction of the insects it lives in, so to give itself an advantage. It works like this. If a male insect has Wolbachia and mates with a female that doesn't, then the eggs she lays won't hatch. If the female has Wolbachia and the male doesn't, she would lay her normal number of eggs. They would all hatch, and all offspring will carry Wolbachia. When two insects that both carry Wolbachia mate, the eggs will hatch, and all offspring will carry Wolbachia. Over a few generations, the number of individuals carrying Wolbachia increases rapidly until nearly all the insects within a population have the bacteria. We wanted to see if we could use Wolbachia as a way to stop dengue transmission. To do this, we first had to move Wolbachia into the mosquito so that it would grow and be passed between generations. Using microscopic needles, we took Wolbachia from the fruit fly and injected it directly into young Aedes aegypti eggs. It took us thousands and thousands of attempts, but finally, we were successful. We then took the mosquitoes that contained Wolbachia and infected them with dengue, and found something really exciting. The virus didn't grow well in the mosquito, and if it can't grow, it can't be passed between people.
Okay, the discovery was great, and after that, many other groups also found more or less the same thing. So that's also possible for dengue, chikungunya, and some work in Plasmodium in Anopheles, also for Zika virus, and as well as yellow fever, which is also was just published very soon. This strategy is part of a not-for-profit initiative called World Mosquito Program, and is now being present in 12 countries, as shown in the map. Before we release mosquitoes, we dialogue with the population to try to build a public acceptance model. We have lots of activities with schools, with community leaders, to really to exchange our information and also, of course, to have approval from them. We also have projects in schools to bring the science not only for Wolbachia, but also related to mosquito control, how they can reduce the breeding sites at their homes. When we get approval, we are ready to produce mosquitoes for releases. For that, we have a biofactory. We produce mosquitoes in a mass-rearing environment, and every week we produce around 4 million eggs and 1.8 adult mosquitoes. To release mosquitoes, we have our team that can just circulate in the car, in the streets. We release every 50 meters, 150 mosquitoes, males and females. And this is done for around 15 weeks. And every week, for instance, in Brazil, we are doing 370 kilometers of driving around releasing mosquitoes. That can also be done by the health agents that help us in areas that are very violent and we cannot enter. They do for us. To monitor whether Wolbachia mosquitoes are being established in the wild, we use these traps. Well, we have a system of 6 motorcycles that run every week. Every day they go to 200 houses or commercial establishments to collect mosquitoes from these traps, and then we will be able to check whether Wolbachia is present there or not in that particular area. So this, the Wolbachia screening is done by molecular techniques, and we, for instance, last month we collected more than 45,000 mosquitoes in those areas with those traps, and we assayed more than 5,000 samples to check whether Wolbachia was present. So when a certain amount of Wolbachia in a particular area goes up, it tends to be sustainable. This graph shows one of our areas, the first areas, pilot areas we've done in Rio de Janeiro. And what you can see here is this blue part here is where when we did the releases, and then Wolbachia percentage going up and just stayed really invaded. The area is being stable for more than 3 years now. In Australia, there are areas that they did this releases for 8 years now, and it's been mosquitoes with Wolbachia is still present in those areas. In Brazil, we are doing work in 2 cities, in Niterói and also Rio de Janeiro. So this is a map of Niterói, and I can show you some results for where we are at the moment. This gray area here is an area that we are not doing any releases, and in the colored area are areas that we are releases or we finished already releases. What you can see in areas like high in green are areas that Wolbachia is above 60%. From in areas that are in yellow, you can see Wolbachia is around 40 to 60%, and below 40% in areas that are in orange. In this city, we are working to cover a population of 271,000 inhabitants. It's a pretty big expansion, what we used to do. Now I show you some results from Rio de Janeiro. Rio de Janeiro, we are working a population of 838,000 people, and you can see also the situation here above 60, here in around 40 to 60, and the other areas below 60, 40, in areas that we are still doing releases, like in blue areas, and areas that we are just investigating the invasion of Wolbachia. I just want to point out that in these areas that we've been working, these two cities, 20% of the areas are favelas, are very complicated areas to work. We deal with violence every day, and it's not easy to work, but is going well. And we work in partnership with the municipality health agents that help us in this process. So an important point of the work is to follow up on cases. So in partnership with the municipality, we've been working this study to see the impact in disease reduction. Of course, this is the end point of what our work. We've been following cases of dengue, historical dengue cases in the city for more than 10 years, and for Zika and chikungunya for the last 3 to 4 years, since the introduction in 2015 in Brazil. I want to show you very preliminary data around chikungunya in Niterói. I don't know if you're aware of chikungunya. Chikungunya is arbovirus. Is very bad in terms of gives a really chronic arthritis in people. People can be really bad for months, sometimes years, with the suffering of arthritis and cannot work. So chikungunya there was epidemics of chikungunya in Niterói in 2018, and this is the area that we did releases, as I mentioned, I showed before. And in the gray area is the area that we are not doing any releases. So I want you to show you the comparison graph of historical following of the cases since 2015. So comparing this purple area with the area in gray, which is our control area. So this is since 2015, what it used to be, and then you don't see practically nothing. And now Wolbachia is being established in this green shaded area here. This is the percentage of Wolbachia around here. And our control area is this area here, the dashed line. And our area of intervention is this green here. So you can see that's already showing up a difference. And we already seen this also for dengue in the same area. If we compare the other area, the brown area now, compared also to the control, you see similar results. In this area, Wolbachia is not so high as the other one, but you can already see a difference in disease burden in the city. We will follow this for the next 5 years. So I'm sure we'll be able to have this year by year. That will be very important for observational analysis. I also want to share with you some data from other countries. The country I want to show is from it's Australia. Australia is not endemic like Brazil. Like when you have a big year of dengue, there are 1,000 cases in the country, the whole country, in the northeast part of the country. But the program was working in this city called Townsville, and worked with 140 people and covered the whole city with Wolbachia mosquitoes. So these graphs, they show historically, since 2002, the imported dengue cases happening in the city. You can see that this every year you have imported dengue cases happening. But when Wolbachia was introduced here when it's in green area, you don't see local acquired dengue cases happening in that city, which is very interesting. So the results for this is that for the pre-Wolbachia intervention period, there were 13 years of local dengue transmission, and when Wolbachia was established, it's been at least 4 seasons that there were no local dengue transmission happening in that particular city. I also want to share with you some preliminary results from Indonesia. Indonesia, they are doing an RCT, which is a randomized control trial, which is the gold standard for epidemiologists to see if that a particular trial is working or not. So this is the map of the city, and you can see here in areas that were randomized to receive Wolbachia in green, or the control areas in white shaded areas here. So this is around 400,000 people that live in this area. And this is the latest results. This is from last year, but this is keeping up the same. This is historical incidence of dengue of areas that Wolbachia is established, like in green, and also the control areas in white and dashed. And you can see here when Wolbachia is established, there is already a difference in the incidence of dengue happening in that particular study. So this is very important. I think we've put together all these results from different countries. It's very important to make the conclusion later on about the impact of on disease reduction. So our next challenge is to really expand the project to other cities in Brazil, but for that we need funding because we're not for profit. Funding to train our personnel, our team, also to produce mosquitoes with the Wolbachia. And I think it's very important to point out that this is a great initiative, and we cannot do by ourselves. It's a group, and also the municipality, and also the people that participate. We have 1,200 volunteers that receive the traps at their houses, and they're important for this to become conclusive. I want to thank the organizers again, the audience here, and I want to thank the funding from the Bill and Melinda Gates Foundation since the beginning, the Ministry of Health in Brazil, the partnership with the municipalities, which is very important, with the schools as well, education and health, and also the team. We have 110 people working in this project in Brazil, and including my wife that's here, and thank you very much.
Thanks a lot. Any questions? Yes, please.
Thank you for this really interesting presentation about this fascinating project. My question is, how challenging is it to convince the public that instead of killing the mosquitoes, you're going to release even more?
Yeah, this is a very important question. Like in the beginning, for everyone we talked about, people say, are you crazy? Why you want to release more mosquitoes? We have enough, and how are you going to do that? It's a process. Of course, when you explain that you're going to release, it's going to be for some time, and eventually that particular region will be protected. They accept. And I'll give you an example. One of the areas, the pilot study areas, it was around 4,000 people. We did all with eggs. There is another way to release mosquitoes, not all the adults, but with the eggs, with a little box. And people keep this box at their houses. And we had around more than 100 distributed in different houses. When the project was over, like we had establishment in that area, we came to remove the boxes. And most of the people said, please don't take it out. We need them. They are protecting us. It was really amazing to see how they really got attached to the mosquitoes that were coming out from the boxes. So it's not because of the engagement, which is very important to, and they suffer a lot with the diseases. If they know that they're going to be protected, I think is the most exciting and most important part.
Okay, this is also not really,
okay.
It's not really a scientific question, but if you really think of an inconvenience of using Wolbachia, what do you think of? You think that there is zero inconvenience? For example, if you are a bird swallowing a mosquito with Wolbachia, nothing happened? When people were starting to use, chemicals or antibiotics, they did not think that resistance would arrive so quickly. So in the case of Wolbachia, the Wolbachia technology, there is absolutely nothing against that?
Oh, I think one thing that comes to my mind when you ask this question is that maybe one day the evolution will come and really this blocking effect will not be there anymore. Could happen, but could take. I think it will take a long time, at least years, decades, maybe to happen. But before that happens, will be greatly have the impact on disease reduction. That's what comes to my mind, and of course, for that we have a biobank, and all the samples since the beginning, mosquitoes in the wild, we kept them for DNA, RNAs, just to study this. If something happens, why this happened, and of course there are other possibilities. In one of the areas you asked about insecticides, our mosquitoes that we were releasing, they were more susceptible to insecticides. And then people started, and the municipality they will start using more insecticides, and then the Wolbachia invasion was not good in that particular area. So we had to do a back-crossing scheme for 8 months to equalize the insecticide resistance of our own material with the field material. So now we do this. We are not releasing super mosquitoes that are completely resistant. They're just the same as what is there in the wild.
I'm sorry for this question, but do you really know the molecular mechanism of the action of Wolbachia on the virus? And is there nothing which would teach you, as we heard from other presentations this afternoon, which you could learn from understanding the mechanism of Wolbachia?
Yeah, so there are many groups that are trying to really pinpoint what is really going on there. There are a few ideas and published work around insect immunity, like Wolbachia was introduced into this mosquito. The immunity is higher, so it will eventually have an effect on the virus. There is a cholesterol story because Wolbachia inside the cell, it uses components of the cell to replicate, and also the virus has to enter in that cell and use the components of that cell to replicate and have the envelope. ROS, reactive oxygen species as well, it's higher when Wolbachia is present. So right now there is a combination, and I believe there's not only one mechanism that is there, which is good, which might take longer for this system, the evolution, to break this the structure of this interaction. But I know that I think it need more science behind in terms of that to really pinpoint what's going on, if that is going to lose sooner or later.
Can you comment a little more about the Plasmodium?
So Plasmodium, there were lots of work on Plasmodium doing transient infection. Like if you inject mosquitoes or Anopheles with Wolbachia, it's able to block. There are some groups working on Plasmodium, on Anopheles, because they found Wolbachia in Plasmodium in some parts in Africa. But apparently, Wolbachia in Anopheles is not as, I would say, like you don't have the CI is not complete, so that's another advantage, because it's not going to be self-sustaining. And also the maternal transmission like in Aedes aegypti, which is 100%, it's not always 100%. So it's also, I think it has to work more on that, but it's very important too. And also, just a final thing is not only one species that transmit malaria, so you would have to put Wolbachia or have a good Wolbachia to block in every single vector, right? If you have only Aegypti, especially in one place, could be is a better solution for arbovirus, I would say. There is another question.
A short question related to the first one on acceptability. Did you encounter some resistance to be in the control areas?
So everything is discussed when we choose an area. Is discussed with the municipality first, and they'll say, "Oh, where can we think we should go?" And they kind of and of course, when we engage people, we say we are doing this at this time. So we hope to get funding from the municipality, for instance, to cover the whole city, but it's not been easy. I think of course, there is a crisis, economic crisis in Brazil. It's complicated.
Question here.
Yes.
Where?
Other techniques have been tested. Other techniques have been tested to fight against mosquitoes, especially for dengue or chikungunya. And I think in Brazil there is also a test with genetically modified mosquitoes. So are you comparing the results? Is there competition between the two methods or cooperation? How does it go between you?
I think one very important thing to say is that we say to the population and also the municipality, they have to continue to do what they do. So we don't block them to spray if needed. We always say to the population that you have to reduce your breeding sites at home. So this is part of our engagement. Brazil, is very big. There are plenty of space for many people to work. So we are not doing interventions the same place that transgenic mosquitoes are being used. So not because we are competitors, but because they are working in different places. But I think this could be used with vector control, should be used with all the vector control strategies and vaccines as well when they become available.
Thank you, Luciano. There is other vectors of dengue, chikungunya, including albopictus. So targeting only Egypti, what happens for the other, which is very good in transmitting chikungunya? So what's your opinion there?
Yeah, so it's a very important question, Annabella. We are aware of this, of course, and like in Brazil, albopictus is not being incriminated as a vector as yet, so it's there. We've been monitoring this and trying to see what's going to happen, but what I can say now, if one day we say, we got rid of the Aedes aegypti vector, and albopictus is there transmitting, we could use a third or put a Wolbachia, which is already done in albopictus, that will block the virus and then start a program with albopictus as well in parallel. That could be done. But at the moment, nobody claims as a vector in Brazil, only aegypti. Fingers crossed.
Is over now. Is it okay? So thanks again for the talk and for this nice discussion.